Hello, everyone. Welcome back to another episode of Omega Matters, where we talk all things omega-3s. I'm Kristina, and with me here is Bill Harris, who will be introducing our guest for today.
Yeah, thank you, Tina. Really happy today to have Dr. Jing Kang with us from Boston University. He's apparently got a snow day there today, so he gets to be at home working. Jing got his PhD, got his MD degree in China, and he then got a PhD at the University of Alberta, and then went on to do postdoctoral work with Alex Leaf at Harvard University, where he remains to this day, now an associate professor in the Department of Medicine at Harvard. And he's also the director of the Laboratory for Lipid Medicine and Technology at Mass General Hospital. It was almost 30 years ago, when I checked PubMed, that he published his first omega-3 paper. That was with Dr. Leaf on, I believe it was cardiac cell membrane biology and omega-3 effects. Since then, he's got over 150 papers on the omega-3. I'm sure he's got way many papers than that, but 150 on omega-3 specifically. We wanted to have him here because he was the inventor and has been the father of one of the most novel research tools in the omega-3 toolbox. And it's not a tool, it's an animal, which is kind of a weird thing, but we'll let him tell you all about it. So Jing, thank you for joining us and welcome.
Thank you. Thank you for having me. Enjoy to share some research we did here in Harvard for the last almost 30 years. Time passes quickly, right?
Yeah.
30 years. I'm 30 this year exactly. So,
yeah, that's great. Let me just start out by asking you to tell us all about the Fat-1 mouse. What is that?
Now, let's first recognize the reality. Many animals, especially mammals, and also include some livestock like poultry and even fish, and cannot synthesize omega-3 fatty acid the whole molecules. So that is why it is so-called essential. I think we understand very well about that. And must be come from must be we must getting from the food. So we create something here, try to get the animal to produce omega-3 without getting from food. That's the technology in nature. That's something that we made the animal capable of produce omega-3 fatty acid. That's the nature of the technology. So we talk about the transgenic Fat-1 mouse model. I think that this is only a part of this technology. Okay, we can create cell, mammalian cell, and livestock, and mouse, and many other animals to do the same thing. So keeping in mind, the thing is make something usually impossible possible now. They are able to produce omega-3 fatty acid without the getting from the diet. So the main thing they do is this technology is get a gene. It's called Fat-1. This gene from the C. elegans, and transport this gene into mammalian cell or in a whole animal, and they can produce omega-3 fatty acid by convert omega-6 to omega-3 actually. And that's the nice thing here. They just not use the carbon hydrogen to synthesize the whole thing. It just simply to convert the existing omega-6 fatty acid to omega-3. It's quite simple, but that's
easy for you to say.
Something that really fantastic thing is that it can change the level of omega-3 fatty acid in the cellular level, in the tissue, in the organ, in the whole body, dramatically. And that's about the that's technology. And the Fat1 mouse, and I think it's simple to understand what is the Fat1 mouse about. And they can do something normally the other mice cannot do it. And normally we have to feed them the omega-3, so enable them to have a higher level of omega-3 fatty acid. Now they can easy to get it, and no matter the diet have the omega-3 or not. Okay.
So what's the advantage in creating a genetic model that makes its own omega-3 versus just feeding the animal omega-3?
Okay, that's a good question. A very fundamental thing. So we want to solve a few important problems. And let me tell you a little bit for generally the omega-3 availability. We know that normally the omega-3 can come from the marine products like seafood and some microalgae and so on. And that's the currently main resources of omega-3 fatty acid. So with the availability of this biotechnology, we can create inland fatty acid without relying on the marine supplies. So we can make the many animals useful for the food, but they have we can enrich the fat acid there, the omega-3 fat acid there, and they can still enjoy the food they like. For example, the steak and the eggs and the bacon, but you can get a lot of omega-3 fat acid there. That's a nice thing for human beings without changing much lifestyle or eating habit, but you're still able to obtain a large amount of the fat acid. That's one thing. And more than that, in terms of the food industry, for the environment issue, we create this technology to produce omega-3, and not just as a convenience, but it's good for human health, for public health, it's good for the environment, and for many things. So I consider our technology, unlike many other genetically modified technology, our technology is to improve the nutrient contents and nutrition value. And I think that this is a very as a promising thing that we can provide such a technology for produce omega-3 fat acid to solve the omega-3 problem with omega-3 availability. And that's the one thing. Now we come back to the research level, and this mouse model can overcome a few very important difficulties or problems that we encounter every day in the lab. And I think everyone working in the field knows very well when we try to modify the fatty acid composition in animal tissue, we have to use different diet or different in the different fatty acid composition. For example, we create a high omega-3, a higher omega-3 or a low omega-3, and we can compare these two different fat acid composition to see which one is better for health. And we usually have to feed animal, right? Corn oil represent the omega-6, and the fish oil represent the omega-3. But we have to understand this is not just our oil, but it's not just the totally pure fatty acid that you get into the cellular level. It's in the diet. And then you feed them, and then you can see how much they can absorb it, to incorporate it, and so on. It's many confounding factors through this process. So we have to make it such a diet. And then so many things there is making very a lot of variables there. So that's the main thing, a main problem for the nutritional study, or for many, especially for the omega-3 fatty acid study, because the omega-3 fatty acid is so unstable, you know that. They can oxidize, they can have so many things can affect the outcome. So that's why we are seeing that the outcome from the omega-3 study, especially for animal study, also in human study, they're so inconsistent and sometimes it's conflicting or a lot of controversy there. We don't know what's really going on. So I think that that's a big issue for the feeding study in terms of we modify the omega-3 fatty acid level in the cell. We talk about the cellular fatty acid, that's important. So with availability of Fat-1 mouse model, because the Fat-1 mouse can convert omega-6 to omega-3. So now we can use that single diet to feed the two group of animals. They eat as identical same thing, but the consequence is the one group have a higher omega-3, one is lower. So we under very nice system there allow us to study a lot of many things for the lipid mediators and for the gene expression and for the many functional level. A lot of things is just based on this difference. We can answer a lot of questions. So that's the first thing we can overcome is the diet, the confounding factor from the diet. That's number one. More than that, a lot of things that we also can allow the research to do a lot to examine many other questions. Normally, we cannot do that in the feeding study. For example, we can look at the development in the effect because we use a mating strategy, allow a mother can have the litter mate be for the Fat-1 or white type in the same litter. So after birth, we can follow, they come from the same mother, but one with the Fat-1 gene, one without, and they eat the same thing, everything the same, identical, but just the fat acid profile. And you can look at the offspring's health condition and the disease development, the sensitivity to some environmental factor and many things we would like to study. And so that we can do that. And we also able to look at something like the gut microbiota. We can use the host of gut microbiota interaction. We can just simply change the gut cells, the fat acid profile without changing the diet. The diet is still the same for the two groups. But one group, they have the high omega-3, one do not. And then you look at what is the gut microbiota in the animal. And that's something we're able to examine. Otherwise, if you feed different diets that they have many other things there, you don't know what's causing the change. Okay.
So you really got two. So your two big motivations, as I see it. One is to create a research model that avoids some of the problems of feeding studies, and the other, which has been, and I want to ask you what you've learned from that a little bit. And the other purpose was to provide a way to get regular foods for humans to have omega-3 naturally. And we want to talk about that too when we get there.
Correct.
Tell us what you've learned from your Fat-1 mouse, just a few of the highlights.
Thank you for your summary. My two things that is one is for technology to produce omega-3. And the second, provide a model for the research that we can, open a new field. We can do a lot of things normally we cannot do, and can clarify a lot of things in the fields. And that's, I think, is quite important. And for the Fat-1 mouse model so far, as I can tell by today, is more than 250 publications there. If you go to search the Fat-1 in the PubMed, and there has to be study different kind of health conditions. I think it's over 50 different things you can see, from the obesity to the longevity to cancer, many things. Also, the people are using the mouse model is worldwide, and I count in more than 20 countries right now. there's something is I didn't expect before, but I think that a lot of people still continue to use this model, and be given this model, this Fat1 also Fat2 to the just laboratory right now. So if anyone want to use it, they can go there to order from there. So based on this, the publication we haven't seen so far, and I think it's interesting that you can see, and many investigators, they use this model. Also, they can use a feeding study to side by side try to validate whether this Fat1 mouse is comparable to the feeding. And also we can use the data to validate that the study done before or the done now, whether this feeding study, which one is true, is real. So I think that's nice that we can see it, and to mainly we can convince the mainstream now the omega-3 is important, and they can do a lot of things there. That's the point. So otherwise, the people always doubt about it. They say, no, this is something else, it's not omega-3 fat and something. So I think that we already addressed many things. And if you look at this publication, simply to change half of the omega-6, not totally, of course, half of the omega-6 to omega-3, they bring the total amount. That's another thing I want to emphasize here. The total amount of the puffer still the same, but just relative amount of omega-6, omega-3 is different. And then you can see a lot of the condition, and from the obesity, fatty liver, to cancer, to mental disease, and brain development, and gut microbiota, and so many things. It's changed. That's why I feel so important this field that, we hope that more people understand the importance of omega-3 for human health. That's same what you guys are doing. I really appreciate that your team is working in human, that we try to get this information together. We can we can see what in the animal, and we can see what in the human. So we haven't find so many things that can be modified. Just change the omega-3 content or change the omega-6, omega-3 relative ratio. So I think that that's something I learned a lot from there. The first thing that they are effective, also affect many things. And secondly, there's some fundamental thing. For example, the chronic information. That's a very important thing. And almost every study to measure the cytokines, and also relate the gene expression. And we find out how they affect the microbiota. This is relatively new. And they affect the lipogenesis. And I view this three things is fundamentally important for the development of a chronic disease. Three things together. And omega-3 fatty acid can profoundly change this three things. Some they more significant than other, or very often there's three things can be affected. And so the important thing is that the many of those chronic diseases we haven't seen right now, like cancer and CVD and diabetes and Alzheimer's disease, they have the same pathology, or they relate to that is three things I mentioned. So omega-3 can significantly change this thing. That's we haven't learned from this many studies that use the Fat1 mouse.
Yeah.
And we can use that for a different level, for the genetic level, from the lipidomics, from the metabolomics, and this is a multi-omics level to document that this effect. I think that is exciting overall. We learn so many.
I can see you're excited.
You see my excitement, right? Excitement, right? Yeah, no, this is the other thing that is going on there. That's another nice thing we can cross this Fat1 mouse model with other disease model, for example, cancer or Alzheimer's disease and many other disease genetic disease model. Then you can follow up from the beginning to the end to see what is changed there, and without we have to get a different diet and they bring the potential confounding fact into the study. So overall, I think that's a lot of things we still can do with this.
Let me ask you: Can you achieve levels in humans equal to what you achieve with your mouse model?
Oh, actually, as I mentioned, many studies that they use the two approaches, and the transgenic mouse model, also the feeding study. And you can see the fat acid composition level is very comparable. Even from lab, they do the different feed a different level for omega-3. You see how much the omega-3 you feed that you can reach to the level in the Fat-1 mouse model. So the study, they get the answer. I think one group in Korea, they published the paper already. They found that if for my body weight, if like a human like 6 kilograms the body weight, if you get a 1 gram per day, you get omega-3 fat acid similar to the Fat-1 mouse model. Oh, so really? It's more than 1 gram, but not this one. It's more than 1 gram EPA, the pure omega-3 fat acid. And so that's very comparable.
So you're saying that, roughly speaking, about 1 gram of EPA DHA per day for a human would get you the levels that you see in your mice?
Yeah, you get
what is that dose? Of course, as you said, you take omega-6 and convert it to omega-3, so you're reducing the omega-6 at the same time.
Yeah.
And can you tell if the effects you're seeing are due to the omega-3 going up or the omega-6 going down?
Wow, that's another very important point, Bill. I think I just point out this model, they use omega-3 as the substrate and to produce omega-3. So when omega-3 up, omega-3 down, and they relatively adjust the level. So today many people have too much omega-6 there, but we cannot reduce omega-6 significantly. We only can push omega-3 in. Okay, we ask them to eat more omega-3, try to get the omega-3 in. If the ratio changes, because it's relies on how much omega-3 we eat. So, and that's why sometimes if we just look at the omega-3 along the amount they take, sometimes we can see the outcome is a little bit different. So what we learned from the Fat-1 mouse model is that if you eat omega-3, in the meantime, you can reduce the certain level for omega-6. That way that you can, like the Fat-1 mouse, decrease one side, also increase another side. Perhaps there's a more effective way. No, I think that we need to prove that in humans if we do a study to validate that. But of course, it's not everyone the same. We depend on the background level of omega-6. I talk about someone if they already have a very high level for omega-6 level there, and if they do the life what we talk about, maybe it's more effective. If someone is already low, and maybe it's not. So I think depend on that. We need to check find out. Okay.
Now let me ask you one more question. Going back to your what you talked about in changing the omega-3 composition of poultry and beef and pork and even fish, right? Have any of those animals been commercialized? Are they available in the marketplace?
Actually, perhaps you should know about it. That's not the technology. That's not a question about the technology. It's a question about the
regulatory,
the policy, the regulatory issue. But I can say this technology is really useful, helpful to the public health. And as I mentioned earlier, unlike many other genetically modified animals that they can make the animal grow faster or resistant to a disease or something else. But our technologies try to increase the nutritional value. It's overcome a big problem that we are facing right now, the whole wide world, because we need that's the nutrients that we cannot just rely on the ocean or rely on the fish, that we can increase the omega-3 in different product lines, and we can allow the people making meat more accessible to the different population. So I think that that's something, even though we have this issue there, that I so confident that that's technology can help. But earlier or later, that I hope that it can come to the market.
Mhm.
But now it's not still. It's not
Not
now.
Yeah.
Yeah, exactly. Well,
Okay.
gosh, I mean, there's so much here, obviously, to talk about.
It's too many things.
Yeah, really. But I think we need to wrap up our talk today. We want to thank you for taking your time to share this
You're welcome.
Fat-1 mouse with people. I'm sure many of them never heard about it, but it's a fascinating development you've done. Congratulations, actually, for thinking about this and doing it.
Thank you.
That's very
Thank
good.
you.
And we wish you well as you continue with this work, and we'll stay in touch.
Thank you. I enjoy talking with you both, and I look forward to more exciting studies. And also for your group, I will tell you more about the recently the few studies from your group that showing something exactly we haven't seen in the other studies, especially related to the immunology, many things.
Great.
Many things.
Thank you. Thank you.
Okay.
So we'll be in touch. Take care. Bye-bye.
All right.
So much.
Bye-bye.